Swayam Prabha. Digital India. Educated India.
Hello, I'm Dr. Haripriya from Sri Ramchandra Medical College and Research Institute, Porur, Chennai. The topic is on histology of muscle tissue. All of you need to know the classification, structure, function, and clinical correlation of muscle tissue. This is a clinical scenario which will be discussed at the end. The patient presents with weakness of the eye muscles, drooping of one or both the eyelids, blurred or double vision, a change in facial expression, difficulty in swallowing, shortness of breath, and impaired speech. Muscle tissue is one of the 4 basic tissue types. The muscle cells have the property of contractility and lie in parallel arrays. They are elongated in the axis of contraction, termed as muscle fibers, and they are mesodermal in origin. The muscle tissue consists of 3 basic elements: the muscle fibers, capillary network, and fibroconnective supporting tissue. Terminology used in muscle tissue. The muscle cell organelles have special names. The cytoplasm is called as the sarcoplasm, the mitochondria, the sarcosomes, the endoplasmic reticulum is called the sarcoplasmic reticulum, and the sarcolemma is a complex of the plasma lemma, its basal lamina, and associated collagen microfibrils. The bulk of sarcoplasm is occupied by myofibrils running in the long axis of the cells. Each myofibril is responsible for the longitudinal striations seen in all muscle fibers and is a bundle of smaller myofilaments, actin and myosin, which are the main contractile proteins. And sarcomere is a linear contractile unit. It's only a segment of the myofibril resulting from the specific arrangement of filaments in a myofibril. The structural classification of muscle. So, 3 types of muscle tissue can be distinguished. The skeletal muscle. It contains bundles of long multinucleated cells with cross striations. The contraction is quick, forceful, and under voluntary control. So, these are the long multinucleated cells with cross striations. The second variety is the cardiac muscle. It has cross striations. They are composed of elongated, often branched cells bound to one another at structures called as intercalated discs, which are unique to the cardiac muscle. The contraction is involuntary, vigorous, or rhythmic. So, these are the muscle cells showing branching, the intercalated discs, and the central nuclei. The third variety is the smooth muscle. Consists of collections of fusiform cells which lack striations and have slow involuntary contractions. So, basically in all types of the muscle, the contraction is caused by the sliding interaction of thick myosin filaments along thin actin filaments. The organization of the skeletal muscle. Connective tissue in the form of thin layers surround and organize the contractile fibers in all 3 types of muscle, but well seen in skeletal muscle. The epimysium is the outermost sheath of the connective tissue which surrounds the entire muscle. The perimysium, it surrounds a fascicle which is a bundle of muscle fibers, and the endomysium surrounds the external lamina of the individual muscle fibers. So, this is the outermost one is the epimysium, this is the perimysium, and then the endomysium. The skeletal muscle is composed of elongated, multinucleated contractile cells called the muscle fibers. So, this is the longitudinal section of a skeletal muscle. The longitudinally sectioned skeletal muscle fibers show striations of alternating light and dark bands. Each fiber is a syncytium with hundreds of nuclei. The nuclei in each fiber are numerous and ovoid, situated peripherally adjacent to the sarcolemma. So, the peripheral nucleus is seen. So, these are the bands, the dark and light bands, and these are the elongated muscle fibers. The ultrastructure of the myofibrils. The sarcoplasm. The myofibrils, they run parallel to the wrong axis of the fiber. The dark bands of the myofibrils are called the A bands, the anisotrophic or bifringent in polarized light microscopy. The light bands are called the I bands, isotrophic, and do not alter the polarized light. So these are the dark bands and the light bands. Each I band is seen to be bisected by a dark transverse line, the Z disc. The functional unit, the sarcomere, extends from one Z disc to another Z disc. The arrangement of myofilaments in a sarcomere. The bands are seen in myofibrils. The A band, it stains more intensely than the I band and shows a central H band that stains less intensely and is less bifringent. In the center of the H band, a slender dark M line may be visible. The Z band is derived from a German word, Zwischenschieb, Zwischen meaning between and Schieb meaning disc. The Z band is a plate between two sarcomeres. The M band is called as the Mittelschieb meaning middle, and the H band is named after Hensen. The molecular structure of the thin and thin filaments, thick and thin filaments. The A and the I banding pattern in sarcomeres is mainly due to the regular arrangement of the thin and the thick and the thin myofilaments. They are composed of the myosin and the F-actin respectively, organized within each myofibril in a symmetric pattern. The thick filament myosin. The thick myosin filaments are 1.5 micrometers long and 15 nanometers wide. They have a smooth central region with sharp projections towards each end called the cross bridges. Each thick filament is formed by a bundle of myosin molecules, each being a rod-like structure of 200 nanometers long and about 2 nanometers in diameter. So these are the myosin filaments made up of several molecules of myosin. So these are the heads of the molecules projecting from the fiber, and they are arranged in a helical pattern. The myosin filament is a bundle of myosin molecules. Two subunits are present in the myosin molecule. The light meromyosin forming most of the shaft and the heavy meromyosin forming the rest of the shaft and the head. The heads protrude from the bundle as cross bridges. Coming to the thin filament, actin. Actin is a thin filament 1 micrometer in length and formed mainly by the F-actin, a filamentous protein that consists of two strands of globular subunits of G-actin, and the two strands are arranged in a helix. The associated with the double helix of actin is a long slender filament of tropomyosin that lies in the groove between the two strands of the F-actin. The tropomyosin molecules lie end to end in the filament. Also present is the troponin in the form of globular units attached to tropomyosin at regular intervals. So this is the disassembled components of the thin filament. So these are the G-actin monomers, the troponin complex, the tropomyosin, and the actin filaments. The A bands contain both thick filaments and the overlapping portions of thin filaments. A band shows the presence of a lighter zone in the center, the H zone. Bisecting the H zone is the M line containing a myosin binding protein, myomysin, and creatinine kinase that holds the thick filaments in place. Sarcoplasmic reticulum and transverse tubule system. In skeletal muscle fibers, the sarcoplasmic reticulum, it surrounds the myofibrils. The sarcolemma has tubular infoldings called the transverse or the T-tubules. These long finger-like invaginations of the cell membrane penetrate deeply into the sarcoplasm and encircle each myofibril near the aligned A and I band boundaries of sarcomeres. Adjacent to each T-tubule are expanded terminal cisternae of sarcoplasmic reticulum. This complex of a T-tubule with two terminal cisternae is called a triad. This triad complex, it allows depolarization of the sarcolemma in a T-tube to affect the sarcoplasmic reticulum and trigger release of calcium ions into cytoplasm around the thick and thin filaments, which initiates the contraction of sarcomeres. The mechanism of contraction. So, sarcomere is the basic unit of the contraction. One end of the thin filament, it runs between and parallel to the thick filaments in the A band for some distance. The other end of the thin filament is attached to the Z line in the I band. During muscular contraction, the thin filaments, they slide between the thick myosin filaments towards the center of the sarcomere, thus bringing the attached Z lines closer, leading to shortening of the contractile unit, that is the sarcomere. It should be noted that during contraction, there is no shortening of the individual thick and thin filaments. The sliding of filaments occurs under the influence of energy released from ATP and calcium ions released from sarcoplasmic reticulum. Coming to the innervation of the muscle, the motor nerves, they branch out within the perimysium, and the terminal twigs, they pass through the endomysium and form synapses with the individual muscle fibers. Each axonal branch, it forms a dilated termination, the neuromuscular junction or the motor end plate. In all synapses, the axon terminal, it contains mitochondria and numerous synaptic vesicles with acetylcholine. The synaptic cleft is present between the axon and the muscle. So, this is a normal neuromuscular junction showing the synaptic cleft, the axon, the terminal, and so these are the synaptic vesicles containing acetylcholine. So, this is the picture seen in myasthenia gravis. The receptors for the acetylcholine are destroyed by autoantibodies. The motor end plate. The single axon and all the muscle fibers in contact with its branches make up a motor unit. The number of motor units and their variable size control the intensity and precision of a muscle contraction. So, what happens at a neuromuscular junction? The nerve action potential, it reaches the motor end plate. The acetylcholine is liberated. It diffuses across the synaptic cleft. It binds to the receptors. Depolarization of the sarcolemma occurs. Muscle action potential is produced, which initiates the contraction cycle. The types of fibers in skeletal muscle fibers. So, we have the red fibers, the white and the intermediate fibers. The red fibers, they are small in diameter, they have more myoglobin and mitochondria, rich blood supply, thick Z disc and complex SR, sarcoplasmic reticulum. The contraction is slow and steady, and they fatigue slowly, example the postural and the limb muscles. The white fibers, they are large in diameter, they have less myoglobin, fewer mitochondria, less rich in blood supply, thin Z disc, and they react quickly with brief forceful contractions and so fatigue rapidly. Example, the extraocular muscles. The intermediate fibers, they have structural and functional properties of both red and white fibers. They are found dispersed among the red and white fibers in the muscles. The muscle spindles and tendon organs. The striated muscles and myotendinous junctions contain sensory receptors. They are acting as proprioceptors, providing the central nervous system with data from the musculoskeletal system. So, among the muscle fascicles are stretch detectors known as muscle spindles. A muscle spindle has few thin muscle fibers filled with nuclei called the intrafusal fibers. The Golgi tendon organs are much smaller encapsulated structures that enclose the sensory axons. This is a H&E stained slide of skeletal muscle longitudinal section seen under high power. The slide demonstrates the histological features of skeletal muscle in longitudinal section. The skeletal muscle fibers are extremely elongated, unbranched cylindrical cells with numerous flattened nuclei located at fairly regular intervals just beneath the sarcolemma. This is the slide of skeletal muscle, a transverse section. Most fibers appear to contain a single nucleus, and the muscle fibers appear polyhedral with flattening of adjacent cells. The myofibrils appear as small dot-like profiles with clear non-fibrillar sarcoplasm lying between and around them. Clear sarcoplasm also lies adjacent to the nuclei. The skeletal muscle fibers are characterized by fibers seen as irregularly round structures with peripheral nuclei. The muscle groups are grouped into fasciculi called as the fields of Cohnheim. The dots within the fibers are myofibrils, which are seen under higher magnification. So, longitudinal sections, so the fibers are easily distinguished as they show characteristic transverse striations. The fibers are long and parallel without branching. Many flat nuclei are placed at the periphery. The muscle fibers are separated by some connective tissue. So, this is the diagnosis for the clinical scenario which was given earlier. It's myasthenia gravis. It's an autoimmune disorder of the neuromuscular junction and occurs due to the failure of the neuromuscular transmission. Postsynaptic acetylcholine receptors are damaged by an autoantibody. As a result, nerve impulses are not transmitted effectively to the muscle, and the patient experiences weakness and rapid muscle fatigue upon voluntary movement. Extraocular and eyelid muscles are affected first, followed by those of the neck and limbs. What's atrophy? So, when the nerve supply of the skeletal muscle is damaged, the individual muscle fibers innervated by that nerve become smaller and thinner, giving the impression of wasting. The opposite process occurs when muscles are exercised over long periods. The muscle bulk in athletes is typically increased, so the process is known as hypertrophy. Muscular dystrophy, there is weakness and wasting of muscles due to a defect in one of the proteins resulting from gene mutations. To summarize, skeletal muscle, they are large, elongated, multinucleated cells, may be up to 1 meter in length. Multiple peripherally situated nuclei are present. They are voluntary, innervated by the alpha motor neurons. They are arranged in fascicles with endomysium, perimysium and epimysium. Cross striations are present due to the organization of the myofibrils, and they have the sliding filament mechanism of contraction. Coming to cardiac muscle. So, the clinical scenario, the patient presents with breathlessness, with activity or even at rest, swelling of the legs, ankles and feet, bloating of the abdomen due to fluid buildup, cough while lying down, difficulty in lying flat to sleep, fatigue, heartbeats that feel rapid, pounding or fluttering, chest discomfort or pressure. The cardiac muscle is found only in the myocardium. It's involuntary, striated and contracts rhythmically and automatically. The cardiac muscle fibers are essentially long cylindrical cells with one or at most two nuclei centrally located within the cells. So, these are the, the fibers there. They are all branching with an intercalated disc and central nucleus. The ends of the fibers are split longitudinally into a smaller number of branches. Between fibers, delicate collagenous tissue similar to the endomysium skeletal muscle supports a rich capillary network. Each cell is about 100 micrometers long and 15 micrometers in diameter. Each cell has a single elongated nucleus centrally located in the sarcoplasm, and it's bounded by the sarcolemma similar to that of the skeletal muscle. The myofibrils are separated by abundant sarcoplasm containing numerous sarcosomes arranged in rows with consequent obvious longitudinal striations. The myofibrils, they diverge around the nuclei and show a pattern of cross striation identical to that of the skeletal muscle with the A, I, Z, H and M bands. The intercalated discs. So they are areas of cell adhesion between adjacent cells. They appear as dark lines in crossing fibers at Z lines in an irregular zigzag or step-like fashion. Their function is to maintain firm cohesion between the adjacent muscle cells and permit rapid spread of contractile stimuli from one cell to another. They are basically specialized cell junctions located at Z lines. The ultrastructure of cardiac muscle. The cell membrane of adjacent myocytes are connected by numerous desmosomes, gap junctions and tight junctions. The desmosomes link the intermediate filaments present in the cytoskeleton of adjacent cells. The actin filaments end in relation to tight junction. The gap junctions allow electrical continuity between the adjacent cells. The cardiac muscle functions as a physiological syncytium. This is a slide of cardiac muscle, longitudinal section, low power. The cardiac muscle fibers, they show branching and a centrally located nucleus. The cross striations in the cardiac muscle fibers closely resemble those seen in the skeletal muscles. The nuclei are visible in the center of each muscle fiber when they are cut in a transverse section. This is again a longitudinal section seen under high power. The branching is well seen with the central nucleus. The highly distinguishing and characteristic feature is the intercalated discs. The cardiac muscle has a vast blood supply. Numerous small blood vessels and capillaries are found in the connective tissue septa and the delicate endomysium between the individual muscle fibers. So the diagnosis for the clinical scenario is cardiomyopathy. It's a disease of the heart muscle that makes it harder for the heart to pump blood to the rest of the body. The cardiomyopathy can lead to heart failure. So all the signs and symptoms of the patient are that of heart failure. The main types of cardiomyopathy include dilated cardiomyopathy, hypertrophic cardiomyopathy and restrictive cardiomyopathy. The cardiac muscle and disease. So atheromas, they lead to narrowing of the vessels and reduction in the blood flow. So a sudden occlusion of a coronary artery may lead to sudden reduction in blood supply to the heart, causing death of the cardiac muscle in a large area of the heart termed a myocardial infarct. In a normal heart, conduction of depolarization for contraction, it runs along the muscle cells themselves. This is interrupted in the case of an area of infarction. A common complication of myocardial infarct is the development of disturbances of the heart rhythm like ventricular fibrillation, and loss of significant portion of the muscle mass from the heart can cause heart failure. Just to summarize, the cardiac muscle shows elongated branching cells joined by intercalated discs, single central nucleus, involuntary muscle, spontaneously contractile with autonomic innervation, shows branching, interconnected cells and acts as a functional syncytium. Cross striations are present due to the organization of the myofibrils, and the sliding filament is a mechanism of contraction. Smooth muscle. Smooth muscle is non-striated, involuntary or plain muscle. It's made up of long spindle-shaped cells having a broad central part and tapering ends. The nucleus is oval or elongated and lies in the central part of the cell. So, the distribution of the smooth muscle. So, it occurs in the walls of hollow viscera, the stomach, intestines, urinary bladder and the uterus. The arteries, veins, bronchi, ureters, vas deferens and uterine tubes show smooth muscle. The muscles that dilate and constrict the pupil. It's seen in the orbitalis, Muller's muscle in the upper eyelid, the prostate, Darto's muscle and erector pili muscles. So the smooth muscle, this is a longitudinal section and a transverse section. It's a drawing of a smooth muscle tissue. The size varies with location. In small blood vessels, they are only up to 20 micrometers long and can reach up to 0.5 millimeters in pregnant uterus. Usually, they are 0.2 millimeters long and 6 micrometers in diameter at the nuclear region. Smooth muscle fibers are spindle-shaped with tapered ends. The cytoplasm of each muscle fiber stains dark. An elongated or ovoid single nucleus is present in the center of each smooth muscle fiber. Occasionally, a longitudinal section can be demonstrated. It's a reflection of the presence of myofibrils running the full length of the fiber. No cross striation or banding is seen. Small dark staining patches may be visible along the sarcolemma and in the sarcoplasm, termed attachment plaques or dense bodies. So this is a slide of ureter, a transverse section of the ureter showing the muscle layer or muscularis layer. The transitional epithelium in an undistended ureter, it shows mucosal folds. The muscularis, it appears to be arranged with the smooth muscle bundles surrounded by connective tissue. The upper ureter has an inner longitudinal smooth muscle layer, a middle circular layer, and a third longitudinal smooth muscle layer is found in the lower third of the ureter. So this is the smooth muscle layer seen in ureter. In the transverse section, only a few cells show the presence of nuclei due to elongation of the cells, but when present, they are round or central in position. In the longitudinal section, the nucleus is elongated and ovoid with rounded cells, stains lightly, and usually has a fine chromatin network. However, in the cells fixed during contraction, they characteristically show a passive flow folded pattern or are pleated in shape. The ultrastructure of the smooth muscle cell. So this is a relaxed smooth muscle cell. The sarcoplasm around the nucleus and particularly at the poles show few sarcosomes, few elements of granular reticulum, and some free ribosomes, a small Golgi apparatus, glycogen, and occasional lipid droplets. The sarcoplasm is occupied mainly by the myofilaments of two types. This is a contracted smooth muscle. The thick filaments vary in length and show cross bridges along the entire length. Thick and thin filaments crisscross the cytoplasm of each cell and are anchored to each cell and to the cell membrane at attachment junctions. The thin filaments are similar to those of the striated muscle but are more numerous with a ratio of 12:1 instead of 6:1 in the striated muscle. So no tubular system is seen in the smooth muscle, but there are elements of sarcoplasmic reticulum and subsarcolemmal caveolae, small pinocytic type of vesicles seen beneath the sarcolemma. The contractile fibers in the smooth muscle stretching between anchoring points on the cell membrane are present. The clinical correlation. So increased bulk of smooth muscle can occur due to a combination of hypertrophy, that is increase in cell size, and hyperplasia, that's increase in the number of smooth muscle cells, since smooth muscle cells retain the ability to divide. To summarize, the smooth muscles show small cells fusiform in shape. They have a single central nucleus. They're involuntary with autonomic innervation. Surrounding a network of collagen is present. No striations. Myofibril meshwork is anchored to dense bodies, and the cells shorten and broaden on contraction. Thank you.